Hello, Assalamu alaikum. Today we are going to study cell organelles and their functions, and this is another series of MCAT. And I'm really sorry for not being uploading and being inactive for some days on the MCAT series because I was not feeling well. But I have resumed it now, and here I am back with another part of the cell which was remaining, in which we will be studying the cell organelles, which are going to be plasma membrane, nucleus, and blah blah blah, whatever. So let's start studying without wasting our time, much of our time. Sorry. So uh, first of all, we have plasma membrane, which just came like floating and swinging on the seesaw. Now over here we have another line known as covers the animal cell. So what is plasma membrane? It is a membrane which is surrounding our beautiful unit of life, which is cell. And this unit of life is surrounded by a plasma membrane, and the most unique thing in this membrane is that it is made up of lipid bilayer. And you can see over here that we have a lipid bilayer, and this lipid bilayer has a function that it has two portions, hydrophobic and hydrophilic. Hydrophilic is the one which loves water, so it is a water lover just like me. And the other part is going to be hydrophobic. Now, which one is hydrophobic? Over here, you can see we have some balls. I don't know if I can zoom in. I cannot zoom in over here. But you can have a look that we have some balls, so many balls over here. Those balls are hydrophilic, while the rope-like structure, thread-like structure, which is down, each ball have two legs over there. You can have a look at it carefully. So these thread-like structures are the hydrophobic, and they do not allow the passage of any molecule across themselves, while they have channel inside which have been shown in the picture in blue color, and these are going to help the transport from this membrane. That's why this membrane is also known as semi-permeable. It can allow the passage of the things it only which cell requires. Okay, so it is a phospholipid bilayer hydrophobic concept have been given, and separates the external environment of the cell with the internal environment. Now we have two environments. Number one is the external environment, and the other is the internal environment. Internal environment is the one having cytoplasm, while the external environment is the one having the tissue fluid. All the tissue fluid is in that section. So there is a division between these both environment with the help of plasma membrane, or else we might be unable to identify which one is the external fluid of the tissue and which one is the internal fluid of the cell if we do not have that plasma membrane. After that, we have a wonderful model presented by Singer and Nicolson. So this is the model of membrane. Over here, I mistakenly made a typo, which is it's membrane, short form for the membrane, MEMM. So fluid mosaic model is a famous model which helps us understand. It is a basic model which helps us understand all the details that the plasma membrane have, that bilayer and the proteins, which I was telling you a little bit earlier, that these are the channels. Those channels are proteins. Over here, the model explains that the bilayer is a liquid on which we have proteins floating over it. So the proteins floating over it are going to be the channels which I just told you behind in this slide. Okay, so let's get back to it. We have two types of protein: internal protein and the peripheral protein. Now the internal or integral proteins, both are the same words, so you can choose what word you love, and it's all about concept. So the internal proteins are those which are inside the membrane. Now we have the lipid bilayer, and after two lipids, at the third lipids, at the space of the lipid, we have the channel. Those sort of channels are known as integral proteins because they are fixing inside the membrane like this. This is the membrane going over there and they fix inside. You can see in the, I guess it's a pink color, and at the right side it's gray, light and dark gray color. These are the integral protein, and at the left side in the image we have purple color. It is known as the peripheral protein, and the peripheral protein are those protein which are hanging outside the membrane. You can see at the right side we have another purple color, and the left one seems like purple, but I don't know which color it is. And over there we also have glycoprotein over the peripheral protein. Peripheral proteins are usually known to perform the function like identification of different cells. Like kidney cells have different peripheral protein, and as the most common example for this concept is that we have different blood groups A, B. So these blood groups are due to the peripheral protein over the RBCs. Okay, let's skip this topic. I have discussed so much over it. Now we have our cell wall. Okay, cell wall. Cell wall is a structure which is covering the animal cell. As I told you, not the animal cell, sorry, the plant cell. As I told you that the animal cell is covered by plasma membrane, while the cell wall is covering our plant cell. Plant cell, the cells of the wood, we can also say. Non-living covering of the cell. This covering is non-living. That is the most specific thing in this, we can say, in this organelle, or in this organelle of the cell. So they can ask this in many of the BCQs in your MCAT, that which one is the non-living part of the cell, and they will give some sort of options in which cell wall is going to be the correct option. Made up of cellulose and polysaccharide, permeable in nature. So it is made up of cellulose. Cellulose, you must remember from your previous classes in your school that cellulose is the component, green color pigmentation inside any leaf. If you see a leaf as green, that's because of chlorophyll. Oh, I was giving you the wrong concept. I'm really sorry about that, and I'm not going to cut this. Let me tell you, from now onwards I am making live, we can say kind of live videos in which there is no editing. I will just make the video and then keep it on my YouTube channel. That's why I may make mistake and correct it, and sometimes I am unable to correct my mistake, so you can correct it in the comment section. Thank you. So I'm really sorry about that. Made up of cellulose. Cellulose is the component which is elastic component which helps the give the cytoskeleton, which makes up the cell wall and permeable in nature. And what else? Provides structural support to plants and helps in water conduction. It helps in the water conduction because it has xylem and phloem inside it. Layers, we have 3 layers. Middle lamella, then we have second primary wall, then we have third secondary wall. The middle lamella, as you can see in the picture, primary wall, and at the bottom and at the upside, we can say upward, is we have middle lamella. So these are some confusing layers which are made up of different components. Middle lamella is the cementing, and after that we have primary wall, which is hemicellulose. It means it's not proper cellulose or pure cellulose. It is a mixture of some other components and cellulose. And then we have secondary wall, which is purely made up of cellulose. Then we have nucleus. Nucleus is the powerhouse, or we can say brain of our cell. This brain was discovered by Robert Brown, and it is double membrane structure. So if you get it in MCQs that these organelles, which one of them is double membrane bounded, then you have to tick at the nucleus. It contains nucleoplasm just like a cell contains cytoplasm, which is also known as karyolymph. Okay, contain genetic structure which is very much responsible for the inheritance, as we usually say that this kid has gone to his dad because he does such certain things which are similar to his dad. So that's why do we say that? Because thanks to genetics and genetic study, which reveals that the inheritance, the similarity from parents in childhood is due to these genetic structures, which sorry, which helps in the production or formation of zygote, and which helps maintain the unique identities in from parents to child. So it contains those nucleus, those chromosomes, and those genes. And let me describe in a little bit more detail. Inside the nucleus, we have nucleolus, okay? And in the nucleolus, we have chromosomes. Chromosomes are something like this, like, no, you cannot see that, but you can see it over here in the image. We have it. So just have a look at the image. We have uh, four four leg four leg something which is known as chromosome, that's written over there. You can have a look at it. I cannot explain the diagrams very much well. So this is chromosomes. After chromosome, when we uncoil the chromosomes, we got the DNA, and then after the DNA we have segments known as solenoid, which is more detail, and after that we have nucleosome. After that they are explaining what are histones, but I have kept histones in the end, while I have then chromosomes, and then we have the DNA. So the segments of DNA are genes, while solenoid and nucleosome are not most important. So let's just skip that part. While the DNA are made up of nucleotide bases, and the histone is the circle ball-like structures which are uncoiled, and then we get our DNA. And in the DNA, segments of DNA are known as gene. And this whole structures of inheritance are known to be made by protein. After that we have chromosomes. Now they just put some detail about chromosomes as well, because that was maybe their most lovely topic or favorite topic. So metacentric, there are types of chromosomes. This is the classification of chromosomes on the basis of their structure. So first type is metacentric, which have equal arms. You can have a look in the diagram. First of all, have a look at the chromosome. We have one circle in between, while two long threads which are thick, and they are known as the arms of our chromosomes, and the ball in between is centromere, and they form up the chromosomes. Now let's discuss the classification, in which we have metacentric, they have equal arms. Then we have submetacentric, they have unequal arms. The arms are not unequal, are not equal, not unequal, not it's they are not equal. We make mistakes, as we are human. We are not that perfect person. Everyone make mistakes. I can make mistake as well. Likewise, I made another mistake in maybe while feeling uncomfortable or whatever it is, because I have never made any live video like this. So I'm not going to edit anything, and that's what making me curious. So after that we have acrocentric or subtelocentric, having one arm small and the other very long. Acrocentric, you can have a look in the picture in the right corner. And then the fourth block is telocentric, centromeres in the end, wherever they are, but they have to be in the end. While in acrocentric, it is very important that the one arm is going to be very small and the other is going to be very long. That's the difference between the acrocentric while submetacentric. Don't get confused that maybe if we think like that, the classification submetacentric says that we have unequal arms. So the arms are unequal in all ways. They can be long, small, 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 or two sides small and the third side long and long. So whatever it can be. While acrocentric has to be one arm small while other very long, not just long, very long. Quite it has to be quite long to make it to stand behind the acrocentric classification. We took so much time over chromosome while this is not much important topic. Let's talk about cytoplasm. What is cytoplasm? The liquid inside the cell in which sorry all cellular activities are performed is known as cytoplasm. Now, this liquid is granular. It has so many cell organelles. So all the organelles we were studying from the start are inside the cytoplasm, while the plasma membrane is surrounding this cytoplasm as well as the we can say the organelles. So we can say the father is going to be the plasma membrane and the cytoplasm is their mother. Okay, and the brain is going to be the nucleus. How weird it is. One family have a one brain. Okay, so a granular liquid containing many important organelles, as I told you, it contains cytosol where all organelles of cell are present. There is one unique thing which is known as cytosol where we have collected or all the organelles are collectively working in the portion. A portion where we have all organelles is known as cytosol. That's what I have been trying to explain over here. And after that we have all the inner fluid of cell including nucleus, nucleoplasm. Inside it is protoplasm, while excluding it we get cytoplasm. Now what I'm trying to explain over there is the difference between protoplasm while the cytoplasm. Protoplasm is actually the all liquids of the cell. It can be liquid inside the nucleus, it can be the liquid inside the cell. While the cytoplasm is just the liquid inside the cell containing cytosol, not the nucleoplasm. Okay, that's the simple difference which I have been trying to explain in the third point. Wow. So we have reached endoplasmic reticulum. So this is another important organelle which are of two types. Number one is going to be the rough endoplasmic reticulum. Number two is the smooth endoplasmic reticulum. Now, what's the difference? Rough endoplasmic reticulum is the type of reticulum which have granulars over it. In short, as the name suggests, sorry, rough is going to be rough in nature or in structure, while the smooth is going to be very smooth, just like my laptop fabric or the mic's structure. So that's what I was trying to explain. After it, what's their function? Rough endoplasmic reticulum forms protein, while the smooth endoplasmic reticulum is forming lipids. Mitochondria, powerhouse of the cell. So we can put it into the digestive system maybe, but I have made lysosome as a digestive system, so I'm not going to name it anything like that. But mitochondria is the powerhouse, we can say generator of the cell. Like it produces ATP, which is adenosine triphosphate, and this is responsible for the cellular activities as well as the activities undergone by the body itself. So the energy currency of a body is adenosine triphosphate, ATP. So it's the money which we call as rupees in Pakistan, money of the body. It is also a double membrane bounded organ. So what was the last in previous organelles, which one was double membrane organelle? That was nucleus, and over here we have mitochondria. It is also a double membrane bounded organelle. It has folds known as cristae. As you can see in the picture, it has folds. These are known as cristae, while it has liquid inside, which is shown in blue color, and it is the matrix of the mitochondria. While we have another thing, it is the only self-replicating organelle as it contains DNA. So all the other organelles are not going to replicate themselves by themselves, while mitochondria is going to be replicating whenever they want. They do not replicate with the cell division, they replicate by themselves within the cell, and they have their own DNA, which is the inheritance material, which I told you the importance of it behind. So let's keep going and quickly finish this lecture. We have Golgi apparatus. Golgi apparatus is also known as dictyosome. So the Golgi apparatus Golgi apparatus are the packagers of the protein, as the protein is formed in two procedures. Number one is transcription and the other is translation. I have a video on this which I'm going to show you in that I button. I don't know what we call it, but in that I button you are going to have the link of my other video, and they are protein packagers. So they package the protein and throw it into the cytoplasm wherever it has to be used. So a protein comes out, when it comes out of the ribosomes, it is in the form of polypeptide chain. Then it goes into the Golgi apparatus. That polypeptide chain is shaped according to the desired requirement. Okay, so that's what it is. Just we have to wind up our lecture. I'm taking too long to explain this. Similar to ribosomes, so don't get confused with it. Yes, they are very similar to ribosome because both the organelles have the same functions in the protein. So sometimes we can get confused into Golgi apparatus or ribosomes, because ribosome is also manufacturing protein, but it is a packager. It just shaped the protein, while we have another thing which is ribosomes, and ribosomes make two units. No, I thought after this I had put the ribosome, but after it's not ribosome. Let's study ribosomes over here. Oh my god, where is ribosomes? Over here. So ribosomes forms two subunits. Over here at the picture you can see the light blue color unit is the small unit, while the orange subunit is the larger unit of the ribosome. They both combine into one messenger RNA, which has a code to make the polypeptide chain, which is desired by the DNA. So this is the messenger RNA which you will be studying in the process of translation and transcription. So wait for it, or just go into the link if you have that topic included in your examinations. So that contains a small unit and a big unit, and it is also responsible for the manufacturing of protein. So this is a long procedure of making of the protein, in which Golgi apparatus is the packager, while the ribosome is the encoder, which reads the messenger RNA and make up the polypeptide chain, which goes into the Golgi apparatus after it. Now we have lysosome. Lysosomes are the cellular digestive system. So whatever debris or whatever dead material inside cell, or unhealthy things, or the unusable things inside the lysosomes, inside the cell, are killed by a lysosome. It can be bacteria, or it can be unusable enzyme, or it can be anything which is harmful for the body, is going to be eaten by the lysosomes and thrown out by exocytosis of which is the process of cell throwing, we can say. Now we have plastids. Plastids are found in plant, not so important. Plant stuffs are not going to be very much important, but it still depends. So I'm not going to make it like you don't have to study that. You also have to learn each and everything, because anything can come. We cannot trust anyone or anyone's statement. So plastids found in plant cell, they are inside the green plants. Chloroplast containing chlorophyll and helps in absorbing light. So the chloroplast is enclosed in a plastid. So plastid act as a container of the chromoplast. Yeah, not chromoplast, or sorry, chlorophyll. I'm making so many mistakes. right now. So chromoplast organelles responsible for giving different colors to flowers. So as we see, we have many different colors in the flower. That's due to the chromoplast. Another small type, these are the types of plastid. We have first chloroplast is also a plastid, chromoplast is also a plastid, leucoplast is also a plastid. While the chloroplast contain chlorophyll, which helps plant to make their own food, while chromoplast give color to our flowers, like red rose give red color to us, leucoplast is colorless and develop in absence of light. If there is a part in the tree where there is absence of light, it start forming leucoplast. Peroxisomes, they help in detoxifying alcohol, majorly found in our kidneys, and plants contain glycoxysomes. Instead of peroxisomes, in plants we have glyoxysomes. It's kind of difficult word. So glyoxysomes instead of peroxisomes, which converts fatty acid into sugar. What does peroxisome in animal do? They detoxify alcohol or any other chemical which they can detoxify. They try their best. If they can, they will. After that, we have, but main function is alcohol. After that, we have glyoxysomes, which are found in plant, converts fatty acids into the sugar, not sugar. They are sugar. Ribosomes, I told you. Cytoskeleton is the skeleton of a cell, just like we have the skeleton in our body, bones, while the cell contains cytoskeleton. Ever thought why don't a cell collapse? So that was quite dramatic, but right now I'm trying to wind up the lecture. So the first line was, ever thought why don't a cell get collapsed while it have only liquid inside it? Whenever you push your arm, the cell may burst out. Some 3 or 4 cells might burst out, if that's the concept. But they do not burst because they have cytoskeleton made up of microfilaments, intermediate filaments, and microtubules. So these are strong filaments. They have elastins and they have certain sort of elasticity which helps them not to burst and sustain pressure. They were cytoskeleton. Now we have centriole, a part of microtubule. No, don't think it's a part of chromosomes, as that is known as centromere. So if you are thinking that it's a part of chromosomes, then no, that was centromere over here, which did on the basis of which we divided chromosomes into 4 parts: submetacentric and metacentric, that all stuff. Over here we are talking about centriole. Centriole is a small organelle which helps all the chromosomes go apart during the cell division, and some goes into the daughter cell while some stays into the parent cells. Or but that's not the concept over here. As we know, one cell divides into 2, so both are going to be the daughter cell, while the parent cell die and it forms 2 more new cells. That's how the division of cell occur. That's why, and in that division, centriole makes the chromosomes go apart, so that chromosomes can go equally 23 into both of the daughter cell, and then they replicate. Oh, I don't know why I go in such details. Hope that it's helping you. Vacuole. Vacuole is the storeroom of the cell, in which we put all the unusable things. That's what you are thinking, because in our houses we use the storeroom as the things we don't need anymore, so we keep them over there. But over here, the things which are very useful for us, and sometimes we don't get those things, like food. Sometimes we have to stay hungry. Don't why we don't die if we don't eat anything the whole day? Because we have food stored into the vacuoles of the cell, and that helps us to stay alive if we don't eat. Why I did not make a thank you slide? I should have a thank you slide. So thank you so much for watching. That's a black screen. I'm really sorry. I'm going to add some text over here. and that's the editing I am going to do and nothing else. Thank you so much for watching again. If you like the video, just hit that like button, you know what it is. And if you do not like the video, I'm really sorry, but you still have to tell me why you don't like the video. Suggest something that I can help you as well, and you can put the thumbs down button over there, and it will be also helpful for me to know. If you don't want to comment, you can just put that thumbs down button, push that button, and color it will be colored as blue, and that's all what I need from you. And you can subscribe below if you want to stay updated with my MCAT series or any other anatomy videos or my physiology videos. More courses will be coming soon, inshallah. Allah Hafiz. That's enough for today. Bye.